Hey everyone, how's it going? Here to talk about our next little subject. We're going to start talking about protein synthesis a little bit. And I think about this a lot. How much detail do we want to go into, and what does the average non-biologist need to know? And so I kind of strike a balance there, but I want to impress upon you the importance of this topic and the things we're going to be talking about. We're basically talking about genetics, and how DNA gets converted to RNA, gets converted to protein, and what does that mean to you? And it's just so incredibly important to biology. And this is where our knowledge is growing so quickly, and it's going to influence so much in biology and so much in your life that it's a very important topic. So let's jump in, shall we? And so what I want to start with in this first lecture is to just talk about some examples of how proteins and what they're used for, and then we're going to talk in more detail in future lectures about specifically how do we build those proteins. And so again, just to reiterate what we've been saying all semester, right? DNA makes RNA, makes protein. That's a central idea to biology. And proteins are, remember, they're big molecules. Their shape is really important. If you change the shape of the protein, you often change its function, and often for the worse. If you change the shape of a protein, most of the time that causes the protein to not be functional. But the important thing is, if you change the shape of the protein, that can be very critical. And so why do we make such a big deal about proteins? We've talked about them before. They're one of the most important classes of molecules we've talked about. What kind of things do proteins do? For example, storage of energy. So albumin inside eggs, right? The yolk of an egg is very high in protein, so the developing embryo has something to eat. So it's storing energy. Like, when you store energy, you tend to store it as fat, but you can store it as protein. Hormones. Hormones are chemical signals that travel around your body. And so something like insulin, right? That's a hormone that signals different cells, and insulin is a protein. Contractile and motor proteins. Muscle. Muscle is mostly protein. And if you eat meat, you know this. Meat is muscle, and meat has lots of protein. We just got done talking about the immune system and antibodies. And antibodies are made of protein. We talked a lot about the cell membrane. And we said the cell membrane is a phospholipid bilayer, but it's got lots and lots of proteins in it. And so, for example, the proteins on the surface of a pathogen that your immune system recognizes, the antigens, those are proteins. There are structural proteins like collagen in your skin. And so it just gives your skin elasticity and toughness, but that's made out of protein. And finally, probably, there's a very, very important function of proteins is that they act as enzymes. And remember that enzymes are catalysts. And catalysts are things that help a reaction go faster, but they themselves are not changed. And that is the importance of enzymes, is that your body is full of enzymes that help the chemical reactions that make you go. They're driven by enzymes. And where do those enzymes come from? They're proteins, and they come from the DNA. And so proteins do just about everything, and about 50% of all living biomass is protein. And so again, understanding how protein's made from DNA, well, that's critical to understanding living things. So that's why we make a big deal out of this. And so we can use examples we've talked about, right? Why do some cells become cancerous? You've got a mutation in the DNA, which changed the resulting protein, And if that protein controls cell division, sometimes that protein now, because of the mutation, the protein changes shape, the protein doesn't work, the cell keeps dividing. That's a tumor. Ultimately, what caused that? That mutation in the DNA that changed the protein. Another example we've already talked about. How do you fight off infections? You've got antibody proteins that perfectly match the proteins on a foreign cell, and that stimulates the entire immune system. Example we want to talk about now in a little bit more detail. Why do different people have different blood types? You're familiar with blood type. Well, let's talk about what is blood type, how is it caused, and this will help again illustrate the importance of protein and its relationship to DNA. So, what's your blood type? A, B, O, AB, or you don't know? If your answer is E, you should find out. You need to know what your blood type is. And so I just ask because I want you to think about what's your blood type. Well, here's a table from your book showing you the frequency of different blood types in the U.S. population. And so you've got, now I just said AB, AB, A or B, AB or O. But of course you also have what's known as the Reese factor. And so O positive is Reese positive and an O negative is Reese negative. And there's probably a lot more complex ways you could categorize each blood types, but these are the most important ones. We're just going to concentrate on the AB system for right now. I'm not going to talk about the Reese factor. But you see that O positive is the most common, followed by A positive, and so on. And so, what controls blood type in humans? Humans have a single gene that controls blood type. And so that's another reason why this is a good example, because it's one gene. Most characteristics in your body are multiple genes and they get complicated. This is one gene, it's on chromosome 9, so we know exactly where it is. But now you're diploid, right? You got one chromosome from your mom and one chromosome from your dad. And so you have 2 copies of every chromosome. So you have 2 number 9 chromosomes in every cell in your body, and each of those has this gene for blood type. And so you have 2 copies of the blood gene type in every one of your cells. And we have 3 different alleles for blood type. And this is how we write them. And so in different situations, we write the alleles in different ways, but this is the common way to write the alleles for blood type. You've got a capital I with a subscript A, that's for the A blood type. A capital I with the subscript B, B blood type, and then a lowercase I. And each of those alleles makes a different protein. And then that's ultimately going to determine your blood type. Now I don't know if we've defined this term allele, so let's make sure we define that and you understand what that is. Remember that a gene is a portion of a chromosome that codes for a protein. And so every chromosome has several genes on it. An allele is a different form of the same gene. And so in this example, everybody has a gene on chromosome 9 which determines their blood type. But there are 3 different forms of this gene, and those forms are called alleles. And so it's by different combinations of alleles that we determine the different characteristics that you have. And in this example, the different combinations of alleles you have will determine what kind of blood type you have. And so you've probably done some of this stuff, and if you've had any kind of biology before, you're familiar with some of this. Okay, and so let's talk about, again, specifically blood type. Remember that all cells are surrounded by this phospholipid bilayer membrane, and it's got molecules on its surface. It's got all these proteins and things on its surface. And those are proteins, but there's also things called glycoproteins and lipoproteins. And so it's a carbohydrate in a protein or a lipid in a protein. And remember that a lot of these proteins are used to recognize self versus non-self. There's lots of proteins, but some of them are used for that self-recognition. Red blood cells, and from here on out, whenever you see RBC, that's red blood cells are no different. they've got that phospholipid bilayer with proteins and other molecules on the surface. And so again, all proteins come from genes on a chromosome. So those glycoproteins that are on the cell surface come from genes too. And different blood types have different glycoproteins on their surface. And so that's how the blood type gene determines blood type. You've got this gene, it's got different forms of, the gene makes protein, that's what genes do, but different forms of the gene make slightly different proteins, and those proteins, in this example, that's what gets stuck on the outside of the red blood cell. That's how you get different blood types. So this, again, calling back to a figure that we've seen before from your book, there's that phospholipid bilayer, you've got proteins sticking through it, and they're showing you sugar chains. That is a glycoprotein. It's got glucose, or sugar, plus protein, a carbohydrate and a protein. So it's just another, it's just a complicated molecule. But these are the types of molecules that allows the body to recognize its own cells. This is that self versus non-self. It's usually glycoproteins. And where do they come from? They come from a gene. And specifically, for the red blood cells, they come from this gene on chromosome number 9. And so each allele makes a slightly different glycoprotein for the surface of the red blood cells. And so the A allele, so it's uppercase I with an A subscript, makes the A-type cell coding. The B allele makes the B-type cell coding. And these, again, are proteins, but they have a slightly different shape. The lowercase I allele makes no cell coding. So that's a gene, it's a form of this blood type gene, but it's mutated in such a way that it does not produce any protein, or it doesn't produce a functioning protein. And so I think how these evolved is that the A allele had a mutation. So the A allele existed, and then it mutated, the DNA changed, and it changed in such a way that it made a slightly different protein, and that became the B allele. And so you change the DNA, that's going to change the RNA that comes from that gene, and you may or may not change the shape of the protein. In this instance, you did change the shape of the protein. It still works, it's just shaped a little differently. Sometimes that happens. Sometimes you mutate the DNA, nothing happens to the protein. Sometimes you mutate the DNA and the protein changes radically. So for example, either the A or the B allele had another mutation, and that created the lowercase I allele. And so this working allele, either A or B, I don't know which one, it mutated in such a way that it doesn't produce a functional protein anymore. So again, this is a perfect example of how, you can't really, you can't predict it, but a mutation can cause either, a working protein, a different protein, a non-working protein. There's all kinds of potential outcomes. And so now we have these three different alleles. A and B make working proteins. They're shaped a little different. I doesn't make any protein. Doesn't work at all. No cell coding is made at all by the I allele. But you got to remember that every human has two of these alleles. And so you have to look at both of them to understand what kind of blood type you're going to be. And so, for example, if you're homozygous for the A allele, homozygous, homo means same, so homozygous means you have two alleles that are the same at that gene locus. If you're homozygous for the A allele, then you have A-type blood. If you're homozygous for the B allele, you have B-type blood. If you're homozygous for the small I allele, that's how you get type O blood. But then you also have heterozygotes. Hetero means different. So heterozygous means you have two different alleles. And so you could have an A and an I, or a B and an I, or an A and a B. A and I also gives you type A blood. B and I, and when I say B and I mean small I. B and small I gives you type B blood. A and small I gives you type A blood. And if you've got an A allele and a B allele, that's how you get type AB blood. And so here's just kind of a table illustrating this. And it's showing you the genotype and kind of a drawing of the red blood cell and the phenotype. Okay, so there's some more vocabulary to make sure that you know. Genotype are the actual alleles that an organism has. So whatever two alleles you have, that's your genotype. The phenotype is the trait created by the genotype. And so if your genotype is two A alleles, your phenotype is type A blood. If your genotype is one A allele and one small I allele, your phenotype is also type A blood, and so on. And you can see the different examples in this table. But now you can understand how we get these different blood types, because it's all about the proteins and the glycoproteins that are on the surface of that red blood cell. If you have a type A allele, you're going to produce a type A protein. If you have a type B allele, you're going to produce a type B protein on your red blood cells. If you have a lowercase I allele, you're not going to produce any protein on the surface. And so that's why a heterozygous person who has an A allele and a small I, they're still type A blood, because they produce, that one allele produces type A protein. And so the red blood cells have type A protein. Their other allele, that small I allele, doesn't produce anything. But you're still, one allele is producing protein, so you're type A. Same thing with B. If you've got an A allele and a B allele, your body produces both and puts them on your red blood cell, so you have both the type A and the type B protein. And remember, the A protein is very similar to the B protein, they're just shaped a little bit differently. If you have two small I alleles, you don't produce any protein for your red blood cells, and your red blood cell has no protein on its surface, glycoprotein. And so here, the red blood cells are covered in the type A cell coating. That's what blood type A means. Same thing for type B. If you've got both the A-shape protein and the B-shape protein on your red blood cells, you have type AB blood. And if you don't have any coating, you've got type O blood. And so this is what I just said. Why do those genotypes create the A phenotype? Because they only create the A glycoprotein. And this is just the same phenomenon that we talked about when we described the immune system. You've got different shapes of proteins on the cell surface that can be identified by your body. It's the same phenomenon in that every cell has got proteins around it. It's just that we have given these proteins specific simple names, A, B, O, because we have a need for it, because we want to know, can we exchange blood among different people, and we need a shorthand to make it easy to describe somebody's blood. And so, we could do this with any cell and any protein, but we don't have a need to. But it's the same idea that you have these proteins on the surface of your cell that help you identify whether that cell belongs to you or not. Okay, so if you have type A blood, who can give you blood? So if you needed a blood transfusion, what type blood could you take if you have type A? Well, of course, you could take other type A's, and you could take type O. Those are the types of blood that you could take. If you've got type AB blood, who can give you blood? Well, type A, you can take type A blood, you can take type B blood, you can take type AB blood, you can take type O blood. You can take any kind of blood if you're blood type AB. And I'm going to back up here for a second. I just want to make sure we're clear. We're not talking about the Reese factor, that, plus, minus. There are other factors, so it's a little more complicated, but you get my point. If you got type O blood, who can give you blood? Well, only type O. If you're type O, you can only take type O. You can't take other blood types. So we had, that's why we came up with these names for these particular proteins on the cell surface, just because we have a need for it. And so again, the different blood types make different glycoproteins on the surface of the red blood cells. That's what they're showing you in this figure from your book. But remember what happens if you have cells in your body that have proteins on them that your body doesn't recognize. Remember when we talked about the immune system? Your body is always looking at other cells to see if it recognizes those cells as self versus non-self. Well, if you get the wrong blood type, you've got the wrong proteins on the surface of your red blood cells. And your body recognizes it and will attack those cells. And so it's the same kind of thing we've been talking about. That's why it's so critical to match blood type whenever you're getting a blood transfusion. But now that you know it in a little bit more detail, you can understand where this comes from. It's coming from these proteins on the surface of these cells. And so if you're a type A person and you're given type B blood, well, that's got the B-shaped protein. Your body's going to recognize that protein does not belong here. If you're a type O, if you've got type O blood, and I give you, say, a type A blood, the type O person has no protein on their red blood cells. Now you're giving them blood, and it's got this protein on it that's got a funny shape. Your body's going to recognize that. It's going to attack it. So that's, this is specifically why it's important to match blood type. And so your immune system will attack anything it recognizes as foreign, which is what we just talked about. And so if you give anybody type O blood, well, there's no protein on type O blood. So there's nothing for the immune system to recognize. I say no carbohydrates, I'm talking about glycoproteins. There's no glycoproteins on that surface, so there's nothing for the immune system to recognize, and so you're fine. And so you can give type O blood to anybody. And so, again, what if you're type A and you're given type B blood? That's a bummer, right? The type B blood has a protein, a foreign protein on it, a foreign glycoprotein, and the type A person's immune system will recognize that and attack it. If you have type AB blood, who can give you blood? Well, you're known as the universal recipient. You can take, if you have type AB blood, which is pretty rare, you can take any blood from anybody. Again, I'm not talking about the Reese factor. There's other things involved. We're just talking about in general here. So if you got type AB blood, of course you can take type AB blood. That's a match. You can take O blood. We just said that O blood has no protein, so you can give it to anybody. Immune system doesn't care. If you have AB and you're given A, well, your body recognizes that A protein. It's not going to attack it. Same thing, if you give that AB person a type B blood, well, their body also makes the type B protein, so they don't attack it. And so both the A and the B factor are expressed. And so the AB blood type is known as the universal recipient. They can take blood from just about anybody. Again, ignoring the Reese factor. If you got type O blood, who can give you blood? Just other type O's, right? If you got any kind of A or B or AB blood that's got proteins on it that your body does not make, and your body's going to recognize those proteins and attack them. But we said that type O can give to anybody. Type O is known as the universal donor. And so type O people can give blood to anyone because they don't produce any proteins. And so that's if you have type O, and if you've ever given blood, you know that your blood is a little bit more valuable than everybody else's blood, because you can just slap that blood into anybody. So, if you got type O blood, you're a superstar. down at the Red Cross. And so this is what I said earlier. Type AB can take blood from anyone, but can only give blood to other type AB. So AB is the universal recipient. Okay. So then we can, we have the kind of classic questions we can ask now about heredity and relationships. And so if a mother has type A blood, her kid has type O blood, and her husband has type B blood, can her husband be the biological father of that kid? So think about it for a second. Well, if you get these type of problems, you always want to start off with what do we know? What just write out what we know for certain. What we know for certain is that the kid has type O blood, and so we know that kid's genotype. That kid must be lowercase I, lowercase I. That's the only way to get type O blood. We know that kid got each of those lowercase I alleles from one from each parent. So we know that one of those alleles came from mom. We know that. Mom has type A blood. So how can mom have type A blood yet still give that lowercase I to her offspring? Well, she must be heterozygous, okay? So now we want to know, can her husband, who has type B blood, can he be the biological father? Well, we know that kid got his other lowercase I from his father. This guy is type B. Can you be type B and still give a lowercase I allele to your offspring? Yes, you can, if you're also heterozygous. And so, yeah, it doesn't prove he's the father, but he can be the father, right? If the mother is heterozygous for the A-type blood, the father is heterozygous for the B-type blood, we know the kid has to be homozygous for the lowercase I allele for O-type to get O-type blood. And so, like I said, we know the kid must be homozygous for the lowercase I allele. We know he got one from mom, and got the other one from dad. Can mom have type A blood and still pass on that lowercase I? Yep, if she's heterozygous. Same thing, can dad have type B blood and pass on that lowercase I? Yep, if he's heterozygous. So this is totally possible. Okay, let's do another example. So you got a mother who's got type O blood, her kid has type A blood, and her husband has type B blood. So in this example, can her husband be the biological father? Always start off with what you know. The mother has type O blood, so you know her genotype. You know she has to be lowercase I, lowercase I. She has to be homozygous for the lowercase I. That's the only way you can get type O blood. Which means that she can only pass a lowercase I allele to her son, to her offspring. And so you know the kid has at least one lowercase I allele, because that's all he can get from his mother. You also know the kid has type A blood. So how can you have type A blood if one of the alleles is that lowercase I? The other one has to be the A allele. So you know the kid's genotype. You know he's heterozygous for the A allele. He's A with lowercase I. And you know he got the lowercase I from his mom. So he had to get that uppercase, that A allele from his dad. Now this woman's husband has type B blood. He does not have that uppercase A allele. He does not have the A allele to pass on to this kid. So there's no way that somebody with type B blood can be the father of this kid. Right? The mother has to be homozygous for lowercase I. The father, we don't know, but we know he's type B. So you know he's either heterozygous with a B allele and a lowercase I, or he's homozygous for the B allele. We know the kid has to be heterozygous for the A allele, because we know he got one lowercase I from his mom. That's all she has. And we know he's type A blood, so his other allele has to be A. But the father does not have that allele. And so that's what this is saying here. Now, again, mutations happen, things happen. Don't go asking your parents their blood type and freaking out if it doesn't match up exactly, because there are times when it gets more complicated than this, so relax. But this is just an illustration of how we can combine what we know about the alleles and the offspring and diploid organisms and all these things to try to predict what your offspring will be from the parents. I'm not trying to start any fights at home here. What we're doing here is an example of what we call Mendelian genetics. And you've probably done these before, these Punnett squares. And it's named after Mendel, Gregor Mendel, who was a monk, who was kind of the first person to really figure out some of these laws of inheritance. And basically this is predicting the offspring's traits based upon the parents' traits. And that's what Mendel did, and that's what we're doing here. You know what the parents' traits are. Now that we know a little bit more about DNA, Mendel didn't know anything about DNA. But when we start to learn about DNA and chromosomes, you realize, oh, that's all based on chromosomes, and so we can learn about the alleles and how the alleles are passed together. That's Mendelian genetics. Or like if breeding, right? If you breed puppies or you breed horses, and if you breed certain colors or certain characteristics together, you sort of can know what to expect in the offspring. It's not always perfectly, you can't predict exactly, but you know what to expect in the offspring. That's Mendelian genetics. But what we've been talking about as far as DNA to RNA to protein, that's molecular genetics. And these days we know how to tie those two together. We can explain Mendelian inheritance with molecular genetics. So not only do we take the parents and then we can sort of predict what's going to happen in the offspring, but we can explain exactly how those things happen in the offspring and what's happening at the molecular level. That's where we're at in the 21st century, and that's where we're really starting to learn a lot about living things. We get a much deeper understanding. So again, here, just another very simple example. Figure from your book at snapdragons type of flower. And this is something called incomplete dominance, which is kind of a Mendelian genetic heredity property. And if you've got a plant that has red flowers and you cross it with a plant that has white flowers, you're going to end up with pink flowers. And you can see that they're giving you the specific genotypes of all these plants, right? So if you're homozygous for the red allele and you cross it with a plant that's homozygous for the white allele, all the offspring will be heterozygous and they'll be pink. And so that predicting how you're going to combine the alleles, that's sort of Mendelian genetics. But then molecular genetics says, well, why? Why does it work this way? And so when you study molecular genetics, you say, oh, that R allele, that big R allele, that's a form of a gene, and it makes a protein, and that protein makes a red pigment. And so if you're homozygous for that big R allele, you have double the dose of that protein, you make a lot of red pigment, and your flowers are red. Now, if you've got homozygous, a white flowered plant that's homozygous for the small R allele, that small R allele has a mutation, and so it does not produce a functioning protein. And so it doesn't produce any pigment because the DNA changed enough to change the protein. The shape of that protein changed enough that it doesn't work anymore. And since the protein doesn't work, it doesn't produce any pigment. And since it doesn't produce any pigment, you get a white flower. And so you see how we're tying the molecular genetics to the Mendelian genetics. But if you cross those together, and now you've got a plant that's got one big R allele and one small R allele, so a heterozygous plant, the big R allele produces a protein that produces a red pigment. The small R allele does not produce a protein that functions, so it doesn't produce any pigment. So you're producing pigment, but you're not producing as much as if you had two of the big R alleles. And so you only produce some pigment, and so your flowers are only tinged with a little bit of red, and they end up being pink. So see how we get a deeper understanding by understanding things at the molecular level. That's where we are at in biology now, and that's why our knowledge is growing very quickly now that we've sort of unlocked a lot of these secrets. Again, think of exactly what happens if you have that big R allele. That is a form of a gene that makes a protein. Now, before, when we talked about blood types, that was a protein that sat on the surface of a cell. That protein that was its job. But you remember I gave you that whole list. Proteins do a lot of things. In this example, the protein that is made is an enzyme. What do enzymes do? Enzymes help chemical reactions. In this case, the chemical reaction is one that creates a red pigment. So if you've got the big R allele, you produce an enzyme that helps to make this chemical reaction that results in red pigment. If you have the small R allele, the DNA's changed a little bit, the RNA's changed a little bit, the protein has changed enough that its shape is messed up and it doesn't work. And so the enzyme does not catalyze a reaction, so the pigment does not get made, and so that, no pigment gets made by that allele. If you've got two of those small R alleles, you don't make any pigment, you got white flowers. If you got one of those small R alleles, you don't produce as much pigment, so you got pink flowers. And so that's what I'm saying right here. And so just, thinking about things at the molecular level helps you get a deeper understanding. And remember again, all proteins are made from genes on the chromosomes. And proteins can do a lot of things. Here's that list again. Proteins are incredibly important. They have a lot of diverse function. Most of the time their function is related to their shape. But one of the most important roles is this role as enzyme. And so those enzymes that are produced from your genes control all those chemical reactions that make you you. You are just a bunch of chemical reactions. And those chemical reactions have to keep going to keep you going. And these enzymes help those reactions go. And where do the enzymes come from? They're proteins made from your genes. So this is how we're linking, chromosomes to actual traits, how we link the genotype to the phenotype. Okay, so that's just an example I wanted to give you. In upcoming ones, we're going to talk about in a little bit more detail how we build these proteins. But as always, if you got questions, you let me know. Otherwise, I'll talk to you later.